What truly happens inside the female body during intimacy is far more complex than most people realize, driven not by assumptions, but by precise biological, neurological, and hormonal responses that shape sensation, emotion, and connection. Intimacy in the female body involves a coordinated interaction between the nervous system, endocrine responses, and vascular changes. During arousal, increased blood flow to genital tissues enhances sensitivity, while neurotransmitters such as dopamine and oxytocin influence pleasure and emotional bonding. Hormonal fluctuations, including estrogen levels, also play a critical role in responsiveness and comfort. Sensory signals travel through complex neural pathways, integrating physical stimulation with psychological perception. Understanding these mechanisms provides a clearer, evidence-based perspective on female experiences during intimacy, highlighting the importance of physiology, emotional state, and overall health in shaping the response. Neurochemical activation during intimacy is primarily driven by the release and regulation of key neurotransmitters and hormones that influence both physical sensation and emotional perception. Dopamine plays a central role in the brain's reward system, increasing feelings of pleasure, motivation, and focus as stimulation occurs. Its release is associated with heightened anticipation and reinforcement of positive sensations. Alongside dopamine, oxytocin is released in significant amounts, often referred to as the bonding hormone, contributing to feelings of trust, relaxation, and emotional closeness. This hormone also reduces stress responses by lowering cortisol levels, allowing the body to remain in a more receptive and calm state. Additionally, serotonin contributes to mood stabilization and overall emotional balance, influencing how pleasurable experiences are processed mentally. The interaction between these chemicals occurs within the central nervous system, particularly involving regions such as the limbic system, which governs emotion and memory. Neural signals triggered by sensory input travel rapidly through these pathways, enhancing both physical sensitivity and psychological awareness. The balance and timing of these neurochemicals vary between individuals, affected by factors such as stress levels, hormonal health, and overall neurological function. Increased genital blood flow is a fundamental physiological response that occurs through complex vascular and autonomic nervous system regulation. When sexual arousal begins, the parasympathetic nervous system becomes more active, leading to vasodilation of blood vessels in the pelvic region. This process allows a greater volume of oxygen-rich blood to enter the genital tissues, increasing sensitivity and responsiveness. The engorgement of erectile tissues is not only a mechanical change, but also a biochemical one, as endothelial cells release signaling molecules such as nitric oxide, which plays a key role in relaxing smooth muscle within blood vessel walls. Nitric oxide signaling is especially important because it directly facilitates increased circulation and tissue expansion. As blood flow rises, nerve endings in the region become more reactive to stimulation due to enhanced oxygen and nutrient delivery. This vascular response also contributes to natural lubrication through transudation of fluid across vaginal walls, which helps reduce friction and maintain comfort. The degree of blood flow can be influenced by cardiovascular health, hormone levels such as estrogen, and emotional state, as stress can activate the sympathetic nervous system and restrict circulation. Over time, repeated activation of this vascular mechanism strengthens tissue responsiveness and contributes to overall reproductive and sexual health by maintaining elasticity and functional integrity of the blood vessels involved. Hormonal influence on arousal is a highly regulated biological process involving multiple endocrine signals that coordinate the body's readiness, sensitivity, and overall physiological response. Among the primary hormones involved, estrogen plays a central role in maintaining genital tissue health, vascular responsiveness, and lubrication capacity. It supports the elasticity of vaginal tissues and enhances blood vessel dilation, which contributes to increased sensitivity. Fluctuations in estrogen levels across different phases of the menstrual cycle can significantly alter responsiveness and comfort levels. Estrogen regulation interacts closely with other hormones to fine-tune these physiological changes. Progesterone also influences arousal by modulating the balance between excitation and inhibition within the nervous system. Higher levels can sometimes reduce sensitivity, while lower levels may allow greater responsiveness depending on the phase of the cycle. Testosterone, although present in smaller amounts, contributes to libido and desire regulation, affecting motivational aspects of sexual response. Testosterone plays a role in energy levels, interest, and overall drive, working in coordination with other endocrine signals rather than acting independently. The hypothalamic pituitary gonadal axis is the central regulatory system controlling these hormonal interactions, ensuring that signals from the brain are translated into appropriate ovarian and systemic responses. Hypothalamic pituitary gonadal axis continuously monitors internal and external cues such as stress, sleep, and emotional state, adjusting hormone secretion accordingly. Cortisol, the primary stress hormone, can suppress reproductive hormones when elevated, reducing physiological readiness and altering sensory perception. These hormonal dynamics do not operate in isolation but are deeply interconnected with neurotransmitter activity and vascular responses, creating a multi-layered system that determines overall experience. Even subtle hormonal shifts can change sensitivity thresholds, emotional processing, and physical comfort levels. Nutritional status, age, and general health further influence endocrine balance, making hormonal regulation a highly individualized process that varies across time and biological conditions. Sensory neural pathways involved in intimacy operate through a highly specialized network of peripheral nerves, spinal cord integration centers, and brain regions responsible for processing touch, emotion, and bodily awareness. When sensory receptors in the genital region are stimulated, they convert mechanical and chemical signals into electrical impulses that travel through afferent nerve fibers toward the central nervous system. These signals are primarily carried by the pudendal nerve, which plays a key role in transmitting tactile and pressure sensations. Pudendal nerve acts as a major communication pathway between the external genital structures and the spinal cord, ensuring rapid and precise signal transmission. Once these impulses reach the spinal cord, they are relayed to higher brain centers, including the thalamus and somatosensory cortex, where physical sensations are interpreted and localized. The brain does not process these signals in isolation. Instead, it integrates them with emotional and cognitive input from the limbic system, which governs emotional response, memory, and reward perception. This integration is why physical stimulation is often experienced alongside emotional and psychological reactions rather than as a purely mechanical sensation. Somatosensory cortex is responsible for mapping the intensity and location of touch, while the limbic system assigns emotional meaning to the experience. Neuroplasticity also plays a role in how these pathways respond over time, as repeated sensory input can strengthen synaptic connections and enhance sensitivity or familiarity depending on context. The spinal cord contains reflexive circuits that can generate immediate physiological responses even before full brain processing occurs, highlighting the layered structure of sensory processing. Additionally, neurotransmitters such as glutamate and gamma-aminobutyric acid regulate excitatory and inhibitory signals within these pathways, maintaining balance between overstimulation and sensory dampening. Emotional state significantly influences neural processing speed and intensity, as stress or anxiety can activate inhibitory pathways that reduce signal transmission efficiency. Conversely, a relaxed physiological state enhances parasympathetic activity, allowing smoother neural communication and heightened sensory awareness. Hormonal interactions further modulate these pathways by altering receptor sensitivity and neurotransmitter availability, creating variability in perception across different biological conditions. The entire system functions as an interconnected loop between body and brain, where sensory input, emotional interpretation, and physiological response continuously influence each other in real time. Psychological emotional connection during intimacy is shaped by complex interactions between cognitive appraisal, emotional regulation systems, and subconscious processing centers in the brain that interpret physical sensations through a personal and relational lens. The brain does not treat physical stimulation as an isolated event. Instead, it integrates sensory input with emotional memory, attachment patterns, and learned experiences stored across neural networks, particularly within regions responsible for emotional salience and social bonding. The limbic system plays a central role in assigning emotional meaning to bodily experiences, with structures such as the amygdala evaluating emotional intensity and potential threat or safety signals, while the hippocampus retrieves contextual memories that influence current perception. The amygdala, hippocampus, and prefrontal cortex work together to regulate emotional responses, where the prefrontal cortex provides cognitive control and interpretation, balancing instinctive emotional reactions with rational awareness and social understanding. Emotional bonding processes are strongly influenced by neurochemical modulation, where hormones like oxytocin enhance trust and perceived closeness, reinforcing emotional security and reducing defensive psychological barriers during intimate experiences. The mirror neuron system contributes to empathy and emotional synchronization, allowing subtle interpretation of emotional cues, body language, and perceived intent, which can intensify emotional resonance between individuals during close physical interaction. Stress and anxiety activate the hypothalamic pituitary adrenal axis, increasing cortisol levels that can interfere with emotional openness, reduce sensory pleasure processing, and shift attention toward protective rather than receptive mental states. Past emotional experiences stored in implicit memory systems can shape present reactions, where positive associations enhance comfort and openness, while negative experiences may trigger subconscious protective responses or emotional withdrawal. Self-perception and body image also influence emotional engagement, as internal narratives about self-worth and acceptance affect how sensory input is interpreted and integrated into emotional experience. Trust formation relies on repeated neural reinforcement of safety signals, gradually reducing emotional inhibition and allowing deeper psychological alignment between emotional state and sensory perception over time. Attention regulation plays a key role, as focused awareness enhances emotional intensity of experience, while divided attention reduces depth of emotional processing and weakens integration between sensory and affective systems. Anticipation and expectation activate dopaminergic reward pathways that prepare the brain for emotional and sensory engagement, shaping how experiences are predicted and emotionally evaluated in real time. Neural connectivity between emotional and sensory systems is strengthened through repeated experiential encoding, where synaptic plasticity enhances communication efficiency between cortical and subcortical brain regions involved in emotional processing and bodily awareness. Autonomic nervous system balance determines how emotional states are physically expressed, with parasympathetic dominance supporting relaxation and emotional receptivity, while sympathetic activation promotes alertness and protective withdrawal responses. Neuroendocrine interactions between brain signaling centers and hormonal feedback loops continuously adjust emotional sensitivity thresholds, influencing how strongly sensory input is interpreted and emotionally prioritized across different physiological conditions. Cognitive interpretation of emotional signals is shaped by individual belief systems, cultural conditioning, and learned behavioral frameworks that define how internal experiences are labeled and consciously understood. Neural integration of emotional and sensory information continues dynamically throughout experience, with feedback loops between cortical evaluation centers and limbic emotional generators constantly refining perception, adjusting intensity, and modulating awareness based on shifting internal states, environmental cues, hormonal fluctuations, and cognitive focus. Allowing continuous adaptation of emotional interpretation without fixed endpoint or termination in processing states' ongoing flow. Individual biological variability is a central factor in how physiological, neurological, hormonal, and emotional responses manifest during intimacy, reflecting the unique interplay of genetics, endocrine balance, nervous system sensitivity, psychological conditioning, and overall health status in each body. No two individuals share identical patterns of response because multiple systems operate simultaneously with differing thresholds, receptor densities, and regulatory feedback mechanisms. Genetic expression influences receptor sensitivity for key neurotransmitters and hormones, meaning that dopamine, serotonin, and oxytocin signaling efficiency can vary significantly between individuals, altering perception of pleasure, emotional bonding, and sensory processing intensity. Variations in estrogen receptor distribution and androgen receptor activity also affect vascular responsiveness, tissue sensitivity, and lubrication efficiency, creating differences in physical readiness and comfort levels across biological profiles. Endocrine fluctuations are not uniform, as baseline hormone levels, circadian rhythms, and metabolic rate contribute to how strongly hormonal signals are expressed and how quickly the body adapts to physiological changes. Nervous system variability further contributes to differences in sensory perception, where some individuals exhibit heightened tactile sensitivity due to increased nerve fiber density or lower sensory thresholds, while others experience more moderated responses due to stronger inhibitory neural regulation. Autonomic nervous system balance, particularly the ratio of sympathetic to parasympathetic activity, determines how efficiently the body transitions between states of alertness and relaxation, influencing both physical and emotional responsiveness in complex ways. Psychological conditioning shaped by early experiences, cultural environment, and learned associations plays a major role in modulating neural processing of sensory input. where cognitive frameworks can amplify or dampen physiological sensations depending on emotional context and expectation patterns. Stress reactivity differs widely among individuals, with variations in hypothalamic pituitary adrenal axis sensitivity affecting cortisol release levels, which in turn influence hormonal balance, vascular function, and neural excitability. Sleep quality, nutritional status, and physical fitness also contribute to systemic variability, as these factors regulate hormone production, neurotransmitter synthesis, and cardiovascular efficiency, all of which directly impact physiological responsiveness. Epigenetic modifications, which alter gene expression without changing DNA structure, can further shape long-term responsiveness patterns based on environmental exposure, lifestyle habits, and stress history, creating adaptive or suppressive effects on biological systems. Age-related changes introduce additional variability, as hormone levels, tissue elasticity, and neural conduction speed gradually shift over time, influencing both physical sensation and emotional interpretation. Immune system activity can also interact with endocrine and nervous systems through cytokine signaling, subtly affecting mood regulation and energy levels, which indirectly influences overall responsiveness. Hydration status and metabolic efficiency determine cellular function and vascular dynamics, impacting how efficiently oxygen and nutrients are delivered to tissues during physiological activation. Even microbiome composition has emerging relevance, as gut-brain axis communication can influence neurotransmitter availability and emotional regulation pathways, adding another layer of individual difference. Psychological traits such as attachment style, emotional resilience, and attentional focus shape how sensory signals are interpreted at a cognitive level, reinforcing the deeply personalized nature of experience. Environmental context, including safety perception, emotional security, and relational trust, modulates neural activity patterns, further amplifying or reducing physiological and emotional responsiveness. The integration of all these variables creates a highly individualized biological system where responses cannot be universally predicted, as each mechanism interacts dynamically with others in continuously shifting feedback loops that define unique experiential patterns without fixed or standardized outcomes.